And Dr. Dr. Judd, thank you so much for coming all the way from Missouri via Zoom to share with us your knowledge and experiences at the Dutch Gap Canal on the James River. We're so excited to learn from you, and I know that you want to encourage people to ask questions, and they can use the chat box to do that. I can kind of curate as we go along, and we can tidy up any other questions at the end. But the floor is yours, Dr. Judd.
Thank you. Thanks, Brahman. I wish I could be there in person. I enjoy getting back to Maryland and DC on occasion, and it's time for another trip. But thanks to you and the Society for the invitation. Um, yeah, today I'm going to transport you to Northern Virginia, but there's going to be some tie-ins to Maryland as well, and then transport you back in time, first 120 or so million years ago, and then back about 150 years ago. So I'll go ahead and share my screen and go into presentation mode. All right, so once again, thanks for the invitation. I'm just so you know about where I'm coming from and all this. I'm originally from Ohio and moved to Maryland for graduate school in 2008 or so. I was there for about 6 years, and I've moved around at different institutions as a postdoc, and now I'm in Missouri as a professor, or assistant professor anyway. But some of my heart is still in the collections in the Smithsonian in Washington D.C. and in the areas around in Virginia and Maryland where we can find plant fossils of Cretaceous age, about 100 to 125 million years ago. So what I'll do tonight is first get you centered. I was going to show this map later, but I always like starting out with a map. It's like reading a good fiction novel. You want to make sure there's a map in the front cover. So take yourselves a little bit south of Maryland. Here's Virginia. So Washington, D.C. and Baltimore all a bit north. I'll show you some more maps. And this is the James River that flows through Richmond and then out into the Chesapeake Bay and ultimately the Atlantic. So we're going to be looking in this area, these kind of reaches and loops, these curves and curls in the James River. And the history of that particular area. A little roadmap for where we're going to go. I'll start by giving you a little bit of background about plant diversity and plant evolution and sort of the interesting questions and mysteries that remain about the evolution of flowering plants and how they relate to other kinds of plants like ferns and conifers, pine trees, things like that. And what we don't know about how it all fits together and how they took over the world in the way they have. Then we'll talk about the geologic setting of the Potomac Group, so the Mesozoic fossil-bearing rocks of the Atlantic coastal plain. And then we'll zoom in on the history of Dutch Gap specifically, and this one particular fossil site that was so transformative for me personally and affected how I look for fossil plants and how I think about the evolution of flowering plants. As we get in through the history, the sort of Civil War history portion, that'll transition to the paleobotany that's gone on, the fossil collecting that's gone on since the Civil War because of the digging of the canal. And then hopefully I can share my my perspective with you on the evolution of flowering plants. I've got here a little trace diagram of the James River as it stretches through that little box that I showed in the previous slide. So the Chesapeake is down here at the bottom, Richmond is off the screen to the north, and I like the curls and meanders of the James River as kind of an analogy of the different things we're going to touch on, because flowering plants and botany in general is pretty different, you might think, from Civil War, and it's just only through a sense of place that these two stories connect. But I think it's an important one to know about. So let's do a little intro botany. I don't know how many botanists we have here, how many plant enthusiasts and gardeners, and how many folks who are here for the Civil War stuff. And just know that plants are green and in the background somewhere. I've got here a magnolia. This is a flowering plant. The flower is front and center here. You can see the big showy white petals, the pink and sort of ivory or pale colored stamens. Those are the pollen producing organs that release pollen that might get caught on the backs or abdomens of an insect that might visit to pollinate. And then in the middle, these little green curls, those are the stigmas. It's the receptive part of the flower where the pollen is delivered either by wind or by insect or whatever the pollinator is. And ultimately, through pollination, the flower, the ovules in the ovary of the flower are fertilized and seeds are produced. And surrounding those seeds are fruits. So this rather complicated structure with attracting organs like petals, with pollinating organs like stamens, and with these receptive stigmas in the center that lead down to the ovules protected inside. This complicated structure is the flower. It's got those successive whorls. There's other things that make flowering plants besides just their flowers. One of them is their distinctive leaves. The leaves of flowering plants are rather different than the leaves of ferns and gymnosperms. And that's one of the ways we can recognize them in the fossil record. Flowers are often ephemeral and often hard to find in the fossil record. They're rarely preserved, although when they are preserved, it's quite spectacular. Leaves and pollen are much more common, and it's those organs that we've used to reconstruct much of what we know about the history of plants on Earth, of land plants anyway. And just to give you the contrast, here's a couple of ferns. These are some interrupted ferns in the genus Osmunda you might recognize. I took this photo in upstate New York, but I'm quite sure that interrupted ferns, you might find them in the woods around Maryland as well. So these don't have flowers, they don't have fruits, they don't have seeds. They reproduce by spores, and those spores germinate in the soil, and new ferns grow from those. There's also the gymnosperms. These are things like conifers and ginkgo. Maybe you've heard of cycads, and there's a few other somewhat obscure groups. Maybe you've heard of ephedra, a plant that's used to make Mormon tea. It was also the source of the compound ephedrine, which was used briefly as a weight loss pill, but has other negative effects, so we don't use it anymore. But most of the time when people think gymnosperm, they think conifers. So here's some taxodium, some bald cypress. These are plants with cones, and inside the cones are the seeds, but there are no fruits. So one of the main differences between flowering plants and other kinds of plants is the presence of the flower structure that we just looked at, and fruits that develop from the flower, from the ovary following pollination of the flower. And it's those fruits that enclose the seeds. So you can always tell you've got a fruit if there are seeds inside of it. So that settles the tomato question. There's seeds in there. Flowering plants are special, not just because we find many of them beautiful, but because they make up most of the diversity of land plants today. So let me take your attention over to this pie chart first. This is all the diversity of land plants today in terms of number of species. There's something on the order of 300 to 400,000 species of flowering plants. Some of the most species-rich groups are things like the sunflower family and the orchid family and the grass family. These are all relatively recent in terms of the evolution of flowering plants. But basically, 9 out of 10 species of all plants on land are flowering plants, and the remainder are going to be in gymnosperms, which is about maybe 8 to 10,000 species of conifers, plus a few others. Bryophytes like mosses and the other bryophytes, liverworts and hornworts. Something like 22,000 maybe species of ferns. Lycophytes, maybe you've heard of ground pine or lycopodium in the woods under a pine forest. There's not many of those. Less than 1% of all diversities in the lycopod or lycophyte group. Maybe you've heard of equisetum or scouring rush. One genus, several species, less than 0.1% of all species of land plants are in the horsetail group. Odds are, if you pick a plant at random, it's going to be a flowering plant. But the world didn't always look that way. This cartoon on the right shows that during the Paleozoic era, way down here at the bottom, we're talking some 400, 420 million years ago, we had the origin of land plants, the evolution of land plants from their green algal ancestors. And between the Devonian, shown here, and this Permian-Triassic boundary, right about here, about 252 million years ago, we see the origin of vascular plants, of trees, so the evolution of woody plants, the evolution of seeds, the earliest conifers, earliest ferns that you would recognize as ferns. Most of the major innovations in plant evolution, the things that allow us to recognize plants as plants, happened during the Paleozoic, during this sort of first phase of land plant evolution. But flowering plants are newcomers in a geological sense. This pink wedge up at the top here says labeled angiosperms, has monocots and dicotyledons. These are different kinds of plants that produce flowers. The monocots are things like palms and grasses and orchids, and the dicots are almost everything else. Notice that they first appear somewhere during the early Cretaceous, somewhere around 130 million years ago, more or less. Exactly when they first occur in rocks, how old the rocks are that you can find fossils in, depends on whether you're looking for pollen or wood or leaves. They show up kind of at different times, but they tell a consistent story. They show up in the Cretaceous, and people have looked really, really hard for older flowering plants, but we're not finding them, or at least the candidates are still debated. The other aspect of this mystery is we don't know who their closest relatives are. So we know they're seed plants because there's seeds in fruit, and we know that conifers and cycads and ginkgo all have seeds. But which one of those is most closely related to flowering plants? That's one of the mysteries of botany still, and there's a lot of people working on it and several different hypotheses. This is a different diagram that shows sort of a similar pattern, except we don't have that sort of balloon shape to it. What you're seeing here is mosses in this green box on the left, and then all the plants that have vascular tissue, basically internal plumbing for water on the right. And then way over here on the right is this green box representing flowering plants. And then way over on the right side of that is this little group called eudicots. Now, if you learned about plants in school, you might have learned about monocots and dicots. Turns out dicots, we don't really use that group in botany the way we used to anymore. So we have this new group called the true dicots, and that's what U means, eudicot, the true dicots. These are just plants where if you cut open the seed and get out the little embryo, it would have two little baby leaves in the seed. So those are the eudicots. About 70% of all plant diversity is in that group. So whereas 90% are flowering plants, most of those are in this eudicot group. And one of the features that we use to recognize them as a group, besides those two little seed leaves, is a feature of their pollen. Their pollen is very distinctive. It's got 3 pores or some variation on that. Whereas all these others, everything else that you see here in that green box is going to have a more simple pollen grain with only one pore or one groove. But the eudicots have 3 or some elaboration of 3 pores. So they're real easy to identify in the fossil record. Pollen is one of the most resistant plant fossils, so it's really useful. So our persistent questions about the evolution of flowering plants and their recent arrival and takeover of the world in terms of species diversity is, which are their closest relatives? What did their ancestors look like? How and why did they spread through Cretaceous ecosystems and ultimately take over? And lastly, where in the world did they originate? Was there some geographic origin that gives us clues about their biology and their evolution? All 3 of these are still a bit open, but they're probably kind of in reverse order in terms of what we know. So we know the most about where they originate, and probably the least, at least I'm the least confident about who their closest living relatives are. My main research interests are in this third question: how and why did they spread through Cretaceous ecosystems? And this is what the fossils at Dutch Gap really shaped my my outlook on this question. So let me transport you now back about 130 million years ago. This is what the world looked like. This is a really nice synthesis from a recent paper by a colleague of mine, Mario Coiro, and colleagues in 2019. And you might be able to make out the red coastline of North America here and much of Asia, South America, and Africa. At this point, the supercontinent of Pangaea has been breaking up. It's in the process of breaking up at this point. You can see the North Atlantic Ocean, where North Africa separated from North America. There's Baltimore somewhere right in there. And these little yellow circles represent primitive angiosperm pollen, the pollen of flowering plants with the one groove, or the one pore. And there are no fossils of this age of the pollen of flowering plants with the three pores or three grooves. What we notice is that where you look for pollen, we find the flowering plants at low latitudes, between 30 degrees north and south. We can find pollen everywhere, but we don't find flowering plants there, just gymnosperms and fern spores and things like that. The column here on the left, as we move through a couple of these slides, gives you a sense of time. So this is the height of this screen is the last 145 million years. And this transition from green to sort of yellow color there, that's 66 million years ago. That's the day the dinosaurs died. And we're going to be thinking about what's happening in this green zone, especially the lower part, the darker green. EK stands for Early Cretaceous. So if we move forward a little bit, the arrow is going to bump up 125 million years ago. Now the primitive pollen type has spread to high latitudes, and the triculpate type has shown up at low latitudes. We can move forward through time a little more, and we see the triculpates have also spread. These eudicot pollen types have also spread to higher latitudes, but more in the southern hemisphere than in the north. By the time we get up here to nearly 100 million years ago, we have both pollen types are globally distributed. So this suggests that over the course of about 20 to 30 million years, flowering plants show up in the fossil record, at least the pollen record, and they spread around the world. So that looks like a takeover story. But that's just from the perspective of pollen. We can also look at fossil woods. This is from a paper that I was on with some colleagues back in 2018. And this graph you're looking at shows time on the x-axis now. So we're starting at about 120 million years ago over here. And we're going to about 66 million years ago. And then I've got a little bracket here. The axis is broken to 0, which is present. So this is millions of years in the past. And the y-axis is the diameter of the fossil woods. How big are the logs? And as you might know, the diameter of a tree trunk is related to the height of the tree. If you want to go measure trees, we often measure diameter at breast height rather than trying to measure the whole height of the tree, because we can sort of substitute one in for the other. So that's all we've done here. And what you can see is that for the early part of the Cretaceous, these first 20 million years here, all of the fossil woods we find that we know are from flowering plants are small, less than 10 centimeters across. They couldn't be much more than shrubs. And we know there are large trees because we have conifers and we have ginkgos, and they're abundant, and they were there for a long time, and they're still there today. So this is just the flowering plant story. And by the time you get to about 90, 95 million years ago, then we start finding larger trees. In this case, the blue triangles and squares are fossils from North America, and the orange ones are from elsewhere in the world. So it's a global database. So it took a while for flowering plants to be represented in the canopy of the forest, even though by 100 million years ago, their pollen could be found all over the world. So there's something going on there, some sort of two-phase spread or takeover, at least. So now we're going to focus in on the Potomac Group and the Maryland, Virginia, Northern Virginia area, because that's where we're going to learn about the fossil record today. And what you're seeing here is an early Cretaceous reconstruction, and maybe 115, 120 million years ago on the left, and about 100 million years ago on the right. So there's a little bit of a difference. Notice the North Atlantic is a little bit bigger. The South Atlantic is a little bit bigger on the right, because we're talking about 10, 20 million years of time passing for those continents to drift apart. Now, during that time, during those 10 to 20 million years, sediment was being deposited on the east coast of North America, specifically on the mid-Atlantic coastal plain. You're probably all familiar with the mid-Atlantic coastal plain, but it's bounded to the west by the fall line and to the east by the Chesapeake Bay or the Atlantic Ocean. And there are several cities that are along the fall line, including Baltimore, Washington, and Richmond. And that's because when European colonists arrived, that was about as far upriver as their boats could go. So it made sense to have a settlement there, up protected away from the tides, but can't go any further. Also, the falls provide a source of hydropower, so it was pretty convenient. We're going to be looking at site number 3 down here. That's going to be Dutch Gap, but notice that there are similar aged fossil sites scattered all along here. So sticking with this green, the rocks of the lower Cretaceous, the sediment where we find fossils from the lower Cretaceous, about 145 to about 100 million years ago, is represented by this green wedge of sediment. And if you go below that wedge, you can find older rocks and older fossils. And if you go above that wedge, you can find younger rocks and younger fossils. And this passive coastal plain margin means that if you walk from the fall line out towards the coast, you're going to be walking past successively younger and younger rocks, or younger and younger sedimentary deposits. So it's way back up here near the fall line that that wedge of Cretaceous rock reaches the surface, and we can collect fossils of this age. If we imagine looking at that same wedge of sediment from the east, sort of back into North America, the green here once again is the Potomac Group. And this is where we're focusing. This is where we can find fossils of early flowering plants in North America, the dark green Potomac Group. So there's Maryland. You can see some recognizable landmarks like Ocean City. And it's way down here in the south that we have the approximate location of Dutch Gap, where the lower Potomac Group here is near the surface. So why the Potomac Group? Well, besides how it's fun to know the natural history of a place where you live and work, the Potomac Group is famous because in 1976 and then in 1977, there were a series of papers published first by Doyle and Hickey and then Hickey and Doyle that looked at the diversity of plant fossils from throughout the Potomac Group, from Maryland, from Virginia, all the sites that had ever been collected. And the question they were trying to answer was, did flowering plants diversify during the Cretaceous, or did they diversify much earlier and then later spread into regions where they might be preserved as fossils so that we could find them? What they found was that as you go from the older rocks at the bottom to the younger rocks at the top, you see the successive appearance of more derived pollen and leaf types in the order you would expect given what we know about the relationships and the diversity of modern plants. You wouldn't expect fossils to show up in that order if they had already evolved somewhere else in the mountains or something, and then later enter the lowlands where we can find fossils. So this was considered a really strong argument that flowering plants diversified in the Cretaceous, which is sort of what we thought already, or many of us thought, it's what we still think today, but it didn't answer the question, who were their closest relatives? That is still an unanswered question. But this work is what made me start by looking at the fossil collections at the Smithsonian in order to introduce myself as a young graduate student to what we think we know about the evolution of flowering plants. And that's why I'm so interested in historical fossil sites from Maryland and Virginia, including Dutch Gap. But you can see some of this diversity. It's kind of interesting. And we'll look at some fossils of some of these. Here you can see the sort of single pore pollen type at the bottom. And then a little bit above that, we see the 3-pored type, and then a bunch of different kinds of variations on that 3-pore structure of pollen. So the lowermost unit of the Potomac Group, that is called lower zone 1, and it's defined by the absence of any of those tricolpate pollen grains, those 3-pored pollen grains that indicate that diverse eudicot clade. So we know there were flowering plants then, but we didn't have that big eudicot group yet. And one of the most interesting sites in this lower zone 1, this lower part of the Patuxent Formation, is the Dutch Gap site. So here's that map I showed you at the beginning. Let's zoom in on the black square. But I'm going to use a historical map for that. This is what it would have looked like in 1863 or so. You can see these different meanders of the James River. And this peninsula right here, around which there's a big reach of the river, today that's called Farrar's Island. and that's where the Henrico Historical Park is. Dutch Gap was cut through this little neck of land between those two, and today you can sail straight up through both of these. There's a new channel was dug here, first Dutch Gap, and then it was extended past Howlett's Farm. This is a remarkable place. This is where Pocahontas was captured and held captive in order to negotiate peace with the Powhatan people of the people of the Powhatan Confederacy. Jamestown is just downstream a little bit. Pocahontas, after she married John Rolfe, she and her son Thomas Rolfe, they grew up here on Verina Farms right across the way from Dutch Gap. This region called the Bermuda Hundreds was established in well, shortly after the Jamestown colony nearly failed, and then John Rolfe came and sort of rescued the colony, established tobacco farming, and sort of saved the Virginia colony. So this little island here, where it says the word Dutch in Dutch Gap, was supposed to be the second settlement after Jamestown, Henrico. But it was that all those plans for a college and a settlement and all that were abandoned after a battle with Native Americans a few years after Pocahontas and her father died. So we're going to be talking about Civil War stuff, but this is also a really important place in colonial history as well. If we move forward to 1864 or so, here's a map of the James River as well. It's drawn a little bit differently, but you can still see Farrar's Island is labeled now, and that's where Dutch Gap is on that neck. Let's see here, at the start of the Civil War, the capital of the Confederacy was moved to Richmond, Virginia, to be closer to the border and sort of secure Virginia's side on the in the Confederacy. And so very early on, the Union was interested in capturing Richmond, and one way to do that would be to bring boats up the James River and take Richmond by water. And in 1861, they tried to do that. Let's see here. In 1861, they tried to do that, and they were really close to taking Richmond. But the general leading the defense of Richmond was injured and replaced. He was replaced with Robert E. Lee. And Robert E. Lee after a few weeks, held off the Union advance, kept Richmond, and for the next 3 or 4 years, one of the goals of the Union Army was to take Richmond. The various armies of the Union were often named after the rivers near where they served. So General Butler was in charge of the Army of the James. So as fighting went on, he was trying to come up with ideas of ways that he could help the Army of the James and the rest of the Union forces take Richmond, either by land or by river. So he hatched a plan in 1863 or 1864 to dig a canal across this little neck separating Farrar's Island from the north side of the river, specifically because this reach, this bend south of Farrar's Island, was both too shallow for their largest gunboats and too close to the Confederate batteries. So you can see this rebel line of entrenchment written here. This is Hewlett's line, and any boats that would try to make it through this shallow reach called Trent's Reach would be within easy shot of the cannons, of the artillery. So by cutting this canal, I thought they could get around all of that and get up to Richmond and take the city. So these are some shots of the Confederate artillery. You can see Confederate soldiers here looking out over the James River, over Trent's Reach there. Another shot up here, you can see the river in the background. This is what General Butler wanted to avoid when sending the boats of the Army of the James upriver. So this is the plan he hatched to dig this canal about 500 feet or so, 500 yards or so let's see, what is it? It's 175 yards, so a little over 500 feet, I think. Um, and all you had to do is make that just wide enough and at least 8 feet deep to get the boats through and avoid the artillery down here south of the river. Yeah. In August of 1864, they started digging the canal. And about a month went by where Union troops, both Union colored troops and white troops, were alternately used to dig the canal. But the work turned out to be really, really hard. And after about a month went by, they needed more labor. The soldiers were needed elsewhere for fighting. And so the solution was to bring in labor from somewhere else. Earlier in 1862, um, Union General Burnside captured Roanoke Island, one of the barrier islands down in North Carolina. These barrier islands were a sort of a safe place for freed slaves or freed enslaved people to sort of wait out the war. They built settlements, they built schools, they lived as freed people, sort of awaiting the end of the war with water between them and those that would seek to re-enslave them. Roanoke Island Freedmen's Colony was one of the most famous of these, as I said, where they built schools and churches and set up a whole town during the war, mostly run and managed by the freedmen. In September of 1864, when it was time to find some additional labor for digging the canal, the Union Army sailed down the James, went to Roanoke Island, and impressed some of those freedmen into labor. And we know about that because two of them wrote a letter to General Butler of the Army of the James asking for their money. Basically, 45 of them were working to help build the settlement on Roanoke Island. And on August 31st, they were told to report to the headquarters to be paid for their work helping the Union Army care for the people on the island. They went according to orders to go get their money. When they got there, they were surrounded by soldiers and they were forced onto a steamboat. When they got on the boat, they were told they were going to be paid. But then once they got on the boat, they told they had to go to Fort Monroe to be paid on the boat. And then they were told they had to go to Dutch Gap to be paid. But in fact, they were just being taken to Dutch Gap to work the canal. So he explains how this all happened to General Butler, which is not the same story that the officers told General Butler about what happened. Then they say the guards went onto the island to take up every man they could, young, old, sick, healthy, caring for sick folks or not, and basically forced into labor. And he notes in the letter that they wanted to go. They wanted the Union to win. They were willing to work for money and willing to work for the Union cause. So he's sort of confused as to why they were impressed into labor in the first place. At the time, contrabands was a term that was used for freed slaves. It was a term used to during the wartime, right? It's as though they were taken property or property that was taken away from the um, members of the Confederacy. But of course, these are people, so that was the term that was used. The two that signed the letter were Ned Baxter and Sam Owens, and as far as I can tell, they were the two most literate of the group. And they explained in the letter that they had only been paid for their work in January and February in Roanoke Island. Remember, this letter was written in September, so they were looking for their money from the last few months and expecting to get paid for the work that they did at Dutch Gap. My understanding is that they were eventually paid, but I'm not sure how much, and I haven't been able to find documentation of their pay. But I think the folks at University of Maryland might know more about that. So some of the photos I'll show you are going to have Black people in them, but some of them are soldiers, members of the troops, and some of them may be the freedmen, and I'm not sure who's who, so I'm not going to try to guess on that. But you'll be able to see progress on the canal. So this is an image drawn from November of 1864. So it started in August 1st. By August 31st, they got the Freedmen and started having them work. And here it is in November, months later, really hard work. You can see this is what will be the canal, and that cliff in the background is the bulkhead that they'll finally blow to let the river through. This is another photo instead of an illustration. You can see in the foreground carts hauling away these Cretaceous muds. These carts are probably full of plant fossils. I wish it wasn't blurry. I could maybe identify some. I always love looking at these old photos. There's a lot of them. And you can kind of see some of the striations in the landscape that are probably real original sedimentary structures from Cretaceous rivers and swamps. You can see the people working. The cloud, the sort of fuzzy part on the right side of the image on the right, is from a shell. So the Confederate artillery is still there across the river. And they're just at the limit of how far those artillery can reach here at the canal. Any closer and the shells would have been a lot more devastating. Finally, a few months later, early January 1865, they were ready to blow the bulkhead and open the Dutch Gap Canal. So they used explosives here to blow up that final cliff. You can see all this debris up in the air as it's drawn for Harper's Magazine. Fortunately, all that heavy Cretaceous sediment and clay went up and came right back down and blocked the canal. So then began the process of digging out all of that mud and debris. In late April 1865, they finally got it open, minimum 4 feet deep, not big enough, not deep enough for the gunboats, but deep enough to pass for some smaller ships. But for those of you who are Civil War history buffs, you might know that late April 1865 is a little late, in addition to being a little shallow for the gunboats. Because April 9th, 1865, Lee surrendered at Appomattox. So it didn't actually help the war effort all that much. But it did help what came later, paleobotany after the war. William Morris Fontaine worked for the U.S. Geological Survey before and after the Civil War, and afterwards he explored the area and found several different sites where he could get plant fossils. This is what it would have looked like in 1885, so about 20 years later. You can still go there today, and you'll go down some stairs on this cliff and go down to the James River and all that. But it would have been much more easy for Fontaine to see the rocks and collect the fossils at that point. He describes some interesting aspects of exploring. He talks about a large tree, some 40 feet long, that was described to him by folks who were excavating the canal back in the 1860s. He talks about these plants called, these plants called Dioenites. There we go. Which had leaves about a yard long. So these would have looked kind of like little palm fronds. I'll show you a picture in just a moment. They were really abundant. Whenever he would find these Dioenites leaves, there'd be nothing else there. But in other beds, other rock layers, he'd find more diverse kinds of plants, conifers and flowering plants and ferns. So he describes a bunch of different plants from both the banks of the Dutch Gap and this unusual ball of clay that was embedded in the canal, probably a rip-up clast. So a flood would have taken sediment from one area and moved it downstream and deposited it in a flood. So you've got both the flora on the of the river near the channel or in a back swamp maybe, and then this other flora represented by a big clay ball. So these are different species. Anyway, he goes on quite a bit in his book about the differences between these plants and the different rock types. Let's show some pictures here. This is one of the ferns that Fontaine found. You might recognize this as an eyelash fern. Skyzia is a genus that grows in the New Jersey pine barrens today. And this is a relative of that genus, Skyzia. Here's another one of Fontaine's ferns in the Smithsonian's collections. one of Fontaine's gymnosperms. So this is a broadleaf conifer. You can see the stem across the middle, and then there are these sort of triangular shaped almost leaves with parallel veins. Here's another conifer. This is one where you've got a main axis. That black line right down the middle is the main stem, and then there are stems branching off of that. And each of those is covered in little scale-like leaves, kind of like you'd see on a redwood. Here's another example of a shoot or a branch of a conifer with some scale-like leaves, and these sort of orangish bodies on the left. Those are cones, seed cones. These aren't very well preserved, and honestly they're kind of worn down because they've been in the collections of the Smithsonian since 1889. So we can look at some fresher stuff. This is one of Fontaine's only flowering plants from the Dutch Gap site. Now, you might not know what you're looking at here, so let me guide your eye a little bit. We've got this bent stem that comes down and then across the screen, and then these sort of reddish splotches on the right side, those are leaves. So it's a little shoot of an herbaceous monocot, something like something like an aroid maybe, a lily, something in that group with more or less parallel veins, but the veins are largely worn off in this specimen that's over 100 years old since it was collected. You can see some conifer branches down here in the lower left, and other bits and pieces scattered that are difficult to identify. So this is one of his only flowering plants. This is what the Dutch Gap site looked like in probably the 90s. So the dark gray stuff at the bottom is where you'd find those dionytes that Fontaine was talking about. And then up above here are different layers deposited in different flood events, where you can find other kinds of plant fossils and rocks and things. I'm going to skip over this one just in the interest of time and take you forward to 1991. This is when there was a field trip with a bunch of paleobotanists visiting the Dutch Gap site to collect some fossils. These are some photos of those dionytes that Fontaine was describing, and they collected them again in the 90s. You can see parts of these fronds. It was difficult to get big blocks, but they got a few nice ones here. This sort of shiny splotch on the left, that's a fossil fern. If you look at the specimen at the Smithsonian today, it's much easier to see, but these sort of black fronds are these cycad-like plants called dionytes. And when you collect the dionytes beds like this, that's almost all you find. Maybe you get lucky and get some ferns, but pretty much you're just looking at these black strips of leaf. Leo Hickey was there at that field trip, leading it in part with some colleagues, and he drew this panel section. So you're looking at kind of two halves of a panorama view of the sediment at Dutch Gap. And these different Roman numerals in green represent different flood events. As sediment was accumulating and the rivers were moving back and forth across the landscape, you get different packages of sediment. And in those different packages, we find different fossils. The lowermost ones, 1 and 2, are where we find those dionytes. This is Leo's, Dr. Hickey's sort of estimate of what the landscape might have looked like, the river meandering through with a conifer canopy, with these cycad-like plants in the back swamp away from the channel, and then ferns scattered throughout with some flowering plants near the river channel, near the levee. And that was sort of the model of where early flowering plants grew based on the work they did in the 70s and later. I'll skip that one. And we'll just go straight to here. So I was looking in the collections of the Potomac Group at the Smithsonian in 2011, 2012, somewhere in there. And I just wanted to get familiar with the work that Dr. Hickey and Dr. Doyle did in the 70s. So I just started looking at everything. And I found this unusual little tiny leaf, shown here on the right, that wasn't described, wasn't identified, but it seemed important to me because it had veins that I could see. It had teeth at the ends of these lobes, and the veins formed closed loops They weren't open and dichotomous like a typical fern. They formed these closed loops, so it seemed worth investigating. So I went on an herbarium crawl. I looked at all the flowering plants I could, as well as the ferns that might look kind of like this in the herbarium at the Smithsonian. I would go in the morning and I'd stay in there till about lunchtime and get through as many as I could. It took several days. Eventually, I'd gone through all of the water lilies, all of the magnolias and their relatives, and several other groups. And eventually, I got into the eudicot group, which I did not expect to get to because there's no triculpate pollen. There's no eudicot pollen at Dutch Gap. But when I saw these on the left here, I knew I was onto something. These are leaves of things like Dutchman's breeches uh, and what's the other one? Corydalis, um, different kinds of herbs that are going to be coming up right now in the springtime in the woods in the next few weeks, all throughout Eastern North America, with really pretty little pink flowers and delicate, almost fern-like leaves. But when you look at them under a microscope and you see their veins, the venation is unlike that of a fern. So, and the smallest ones have veins very similar to the fossil. On the right here, just to show you that I did check all the ferns to make sure there were no lookalikes hiding in some other kind of plant group. And I was able to rule out all these alternatives. Up at the top here, you've got maidenhair fern. This lobe down here is an aquatic fern called Ceratopterus. Grows down in the southern US in ponds and wetlands. And this one on the right here is a species called, or a genus called Tectaria. It grows in tropical rainforests and it's got lobed leaves and veins that make complete loops. But all these little black dots, that's where the spores are produced. That's how you know it's not a flowering plant. So after doing all this comparison, I began describing the fossil, and in the process of describing, I had to give it a name. So I named it Potomacapnos, which means poppy, basically, or fumitory of the Potomac Group. And then this epithet, this second part of the species name, you know, for humans, it's Homo sapiens, sapiens meaning knowing or knowledge. Apeleutheron is the Greek for freedmen's. So it's a possessive, it's an unusual name, but it's a possessive form of the word apeleutheros, and it means freedmen's poppy of the Potomac. So all this work that had been done by Fontaine and Leo and Jim Doyle, Leo Hickey and Jim Doyle, and Gary Upchurch and others who had come later, all the way up to me and some of my colleagues were still working on this stuff. All that work was made possible by the digging mostly of these freedmen who were impressed into labor. So I thought it was appropriate to name at least one of the fossils out of there after their efforts. But it led to a mystery. Let me do a little time check here. I'm going to wrap up here in just a moment. It led to a mystery of why are we finding leaves that look like eudicots when I was expecting to find leaves like these? Broad leaves, vaguely magnolia-ish. That's what I thought I was going to find. But with this new search image, I started seeing these little loby things everywhere. This is one I described also from Dutch Gap, but it also occurs in Maryland at Federal Hill and in downtown Baltimore. Several different examples of these little loby leaves on herbaceous stems. Lots of species. Fontaine actually found these, but he thought they were ferns, misidentified them. Here's a few more specimens of the same thing. These are in the Dionysus beds. They're small, and so they were missed. This is a reconstruction I did with a colleague of this plant. You can see the leaves there coming off of this little herbaceous stem. And in the background, we've got a baby Tenontosaurus, an iguanodon relative, to give you a sense of scale. So this is the summary of what it's like in the Potomac Group, the successive appearance of different leaf types. Why did we miss these little things? Because they're little. Those are the two species I've just shown you, Potomacapnos on the right and Fairlingtonia on the left. And there's others that I haven't written about or described yet. These are also fossils that Fontaine thought were ferns, but in fact are not. They're early flowering plants, probably angiosperms, maybe eudicots. So I thought they were eudicots, and I thought that was sort of it. And the reason we didn't have the pollen is because they were insect pollinated, so they didn't make much pollen, so it's unlikely to be preserved, unlikely to be found, unlikely to be identified. But this isn't the only place where these kinds of fossils predate the earliest occurrence of the pollen you would expect to find with them. One example is in Argentina. Just last year, Coiro et al. Found this thing, which was also previously misidentified, and he showed it's a flowering plant, similar to Potomacapnos and others. Um, but it has some extra features preserved that weren't preserved on the ones at Dutch Gap that aren't very consistent with this eudicot group. So maybe it's actually some other plant group, still a flowering plant that evolved to have leaves that look similar, but didn't have the right pollen. It's sort of an ongoing debate. We're not really sure what to make of it yet, or at least I'm not. So with this new search image, whether they're eudicots or some early relative, I spent more of my time working in Wyoming, in the Cloverly Formation, which is about the same age as the Potomac Group. So what I'll do is I'll end with a few photos and a little summary of how that Potomac Group stuff affected my work out here in Wyoming. This area is famous for dinosaurs, but there's plant fossils too. Here are some of those plant fossils. Once again, herbaceous things, small leaves. These scale bars are a centimeter or less in all cases. Lobed net veins, just like you would expect to see on an herb. This one's really complicated. This one's got 3 leaflets. This one, it turns out, has some of the same features that are on the Argentina specimen I told you about, but it occurs with tricolpate pollen. So there's still some stuff to work out there. Some other examples of little herbs. They're all over the place there. Generally rare, hard to find. You look at a lot of ferns and a lot of sticks and conifers before you find these things. This is what I mean when I say they're rare. The y-axis here is the number of sites, how many holes in the ground we dug looking for fossils. And the columns are the different species that we can recognize. And the flowering plants in red, look at this, most of the flowering plant species are herbaceous, first of all, and most of them have been found only at one hole in the ground in northern Wyoming and nowhere else. So they were generally rare, both locally in terms of the number of leaves you find and in terms of how many places you can find them. This is not the pattern you would expect if you thought that the reason flowering plants took over is because they outcompeted the ferns and gymnosperms. This is the pattern you would expect if they diversified in a way that allowed them to avoid competition with these incumbent Mesozoic plants like conifers and ferns, and sort of infiltrate all of the world's ecosystems before ever taking over the canopy as large trees. That's the classic model. So here's that angiosperms on the right here, shows their diversification. And there's this decline in gymnosperms. So the traditional model, I've seen it even this past year in some publications, is that flowering plants took over because they outcompeted and drove the gymnosperms to extinction, at least some of them. But that's not what the first 20, 30, 40 million years were like of flowering plant evolution. That's a long time. So I'll wrap it up here. We're just after 7 or 7 my time. The Dutch Gap site provides a window into the early history of flowering plants some 115, 120 million years ago. And access to that evidence and the collections that are now in the museum at the Smithsonian are largely a consequence of the work of freedmen impressed into labor by the Union Army during the Civil War. In describing Potomacapnos, I developed a new search image for small lobed flowering plant leaves instead of those big broad magnolia type leaves. And that has influenced my research ever since, especially out in Wyoming. And what I think we learned from this is that for the first 20, 30 million years, these plants were diversifying mostly as small herbs, and they weren't out competing or displacing the Mesozoic plants. They were just integrating with those ecosystems. And it wasn't until much later that we see the transition to a world dominated by flowering plants in terms of cover and canopy of forests. With that, I'll acknowledge Leo Hickey up here on the right, who unfortunately passed away while the Potomacapnos paper was in publication. or in the process of being published. I'll thank Scott Wing, my graduate advisor at the Smithsonian, who's done a lot of really interesting work, as well as my other thesis committee members as a graduate student, and Jim Doyle and Gary Upchurch, who've been really helpful for discussion and thinking through these things as the years have gone by. I'll also thank Stephen Miller at the Freedmen and Southern Society Project at the University of Maryland, who was able to track down a copy of the letter written by Ned Baxter and Sam Owens asking for their pay for their work, and the Library of Congress for those images of the artillery and things like that, and a couple books that help us try to piece together what happened there at Dutch Gap. I'll stop there, take any questions if we have time. I maybe went a little over, but I'm not going anywhere. And yeah, I'll open it up.
Thanks. Thank you, Dr. Judd. Can you stop sharing? We can all come back together. That was phenomenal. I loved so many aspects of the presentation because it was like the river, the adventure.
Yeah.
I love that. But it also talked about the importance of these collections that we have that have been done over the years. I mean, I'm thinking, what do we have? We might have some of these in our collections down here in Overley. So that's quite exciting to maybe use your new search engine to look at them with the new eyes to see what we have. We do have some questions, and let's see. What are some questions here? Mason, you wanna unmute and ask?
Sure. Awesome presentation. Loved it. And great job naming that species after the Freedmen. They definitely deserved it after all this time. So are there any vertebrate fossils known from Dutch Gap? And is there any ongoing research at the site?
As far as I know, the only vertebrate fossil is a fish. And that fish is at the Smithsonian, and it's surprisingly similar to fish at a similar age site in northern Brazil. So I think there's been a little bit of comparison of the different fish that would have lived around that ancient seaway that would have had South America on the southern part and North America on the northern part, and also Africa. But I think that's the only vertebrate fossil. Jim Doyle and Gary Upchurch reported finding remains or evidence of insects. And there's probably evidence of insect damage on some of the leaves, but I think the fish is all we have in terms of vertebrates. But there are other bones from the Potomac Group throughout the area where there's great footprints. There's an exceptional footprint site that was published just a couple of years ago. I encourage you to look it up. Footprints, Potomac Group, dinosaur. You'll find it. It's incredible.
Have Shireen is, how do you find, isolate, and analyze the pollen specimens?
Yeah. I can't really do that where I am now because it requires really strong acids. So we have to do what's called acid maceration. We take a sample of rock, make sure it's clean, make sure there's no modern dirt or dust on it. We have to separate out the really nice clean sample of Cretaceous rock. And then that goes through a series of acid washes that gets rid of the sand, the mica, the clay, all the different minerals and sediment particles that are in there. But it doesn't affect organic material like pollen and spores. So by the end of going through all that process, you end up with a solution that's almost pure pollen and spores and other debris of plant. Nowadays, I send it to a lab in Canada and they do it for me for a fee. But at the Smithsonian, there's paleontologists who will do those preparations. Good question.
I was also interested because you mentioned the plants being found in the Baltimore Federal Hill area. Do you know more about that?
There is a famous Potomac Group site in Fontaine's book called Federal Hill, and I think he collected while they were there's like while they were making the hill, right, and the little path around it with the flag and all that. There's other sites that are, I'm not sure exactly where they were, but they were definitely somewhere in downtown Baltimore, maybe a construction site, that sort of thing. But the records aren't always that great.
And let's see, Jonathan wants to know, what do you think drove the eventual rise to the dominance of the angiosperms?
Yeah, I think that's going to be a two-part answer. I think that there were climatic changes during the late Cretaceous. So we're talking 70 million years ago, something like that, that uh, that started getting flowering plants to be more important in the forest canopies and things like that. Um, but we don't see real angiosperm dominated rainforests without conifers until after the extinction of the dinosaurs. So I think that had a big influence in terms of sort of wiping the slate clean over large areas of the earth. And then they were able to come back in on a level playing field. Whereas during the Cretaceous, other plants were incumbent and they had sort of the home field advantage, you could say.
Any other questions for Dr. Judd? Oh, Mason is letting us know that when it opens back up Dinosaur Park in Laurel, you can go see the Potomac Group. And you
And
can take a piece of wood.
yeah, they've got lots of wood fossils and cones of some of those conifers that would have dominated the forest canopies.
And are these sites open for continued fossil hunting or no?
I blew right past that on the Dutch Gap site. I visited the Dutch Gap site in 2013 and it's covered in riprap. It's totally protected from erosion. There's a few spots down near the water at low tide where you could maybe poke around. I had permission. I don't actually know what the rules are now, but I let them know I was coming and all of that, and didn't actually collect anything. We looked at the rocks and thought about it, but we didn't actually collect much because the real good sites are all covered. There's sort of secret little spots around Maryland and Virginia where if behind shopping developments and stuff where you can still find pockets of Potomac Group and look for it. But as far as I know, the only one that's sort of officially open as a place you can go is the Dinosaur Park in Laurel.
Any other questions for Dr. Judd? I feel smarter. I don't know about you, but this has been wonderful. Thank you so much. And now your homework for everybody is to share what you've learned with other people to make them smarter. So spread the knowledge around. And you are welcome to come back anytime, Dr. Judd, to give us more of your knowledge and share with us anytime.
Sounds good.
And come to our facility and maybe look at some of our collections to see what
Next
we
time I'm in the DC area, I will be sure to visit. I look forward to it.
That's wonderful. Everybody, thanks for joining us. I hope you come back and learn with us some more every Thursday at 7. There's something to learn here at the Natural History Society of Maryland. Join us. Stay safe and stay curious, everybody. Take care.
All right.
Thank you so much.
Yeah. Happy